Good morning, everyone. My name is Sharon Begley. I'm the senior science writer at STAT. Thank you very much to the organizers for asking me to play Charlie Rose for an hour or so this morning. My esteemed guest is Mike Snyder. Mike is professor and chair of genetics at Stanford University, where he is also director of the Center for Genomics and Personalized Medicine. Mike received his PhD at Caltech. He did his postdoc work at Stanford, where his lab has developed many genomic and proteomic technologies. So we'll talk about some of those in general, but the main reason we're up here in front of you this morning is that about 4 years ago, Mike and his colleagues created an integrative personal omics profile, or iPOP, of Snyder himself. It has since become known as the Snyderome. It's an ongoing project consisting of, among other things, Mike's genome sequence, transcriptome data, proteomic data, metabolomics data. There was so much Mike that one wag described it as a narcissisome. But the Snyderome, originally published in Cell in 2012, is not about ego, or so Mike says. Instead, it is a sort of demonstration project intended to show how much we can learn by studying individuals over time, not only by comparing one ohm to another at a single point in time. So, Mike, for those in the audience who have not been following the work for 4 years plus, I wonder if you could just start off by telling us what in the world possessed you to undertake this, and why yourself as opposed to some lab rat or lab rats in Palo Alto.
Sure. I think what possessed me to start this project was when I moved from Yale to Stanford, it hit upon me, well, we had been talking about it, so I was at Yale for 23 years, and we were doing a lot of high throughput technologies, meaning we make thousands of measurements in almost everything we do. And when I moved from Yale to Stanford, I enrolled in a new health plan, like one does, and they take your blood and they give you back a report with 15 things. And I thought this was ridiculous in today's technology where you can make thousands or if not tens of thousands of measurements off tiny little drops. Why are we being so imprecise? Why are we being so, if you will, cave person-like in our analysis of people's samples when we could do so much more? And the reason we chose me was twofold. One is I was accessible. I didn't have to worry about, if you want to give longitudinal samples, I knew I could always give them from me, as opposed to calling someone in. And the other was something that surprised me. When I moved to Stanford, which was in 2009, they wanted to actually let medical students and first-year graduate students genotype themselves. It's an optional class. It was optional to actually genotype yourself within the class. And it created a big brouhaha with the basic scientists being 100% in favor and the clinicians being 100% against. Okay, they were afraid that if students genotype themselves, they might learn something awful, and they weren't prepared to handle this. And I just thought this was ridiculous. In an age where genome sequencing, there were only 2 genomes out at the time, but it was clear where the technology was going. And it just seemed like we had to get people on board and comfortable with this technology. So I wasn't going to go out and grab some random person. It's a lot easier to grab me to start this study on, simply to show we can get this going. Didn't have to worry about suing myself, anything like that. So that's how we wound up choosing me. Kind of a long story, but.
So 15 measurements in your standard, physical blood test. Give us a sense of the scale of your project. How many measurements and how many kind of things?
Well, so from the biochemical side, we measure as many molecules as possible out of my blood and out of my urine. So that means all my proteins, my RNA, my metabolites, my antibodies. I do 5 different microbiomes. So the most obvious ones are your stool microbiome, your nasal, skin, urine, and actually oral microbiome. What else do? We do my modified DNA called your methylome. And we're starting to add on some more cellular assays. We actually measure detailed cellular composition with something called CyTOF. It's a very updated imaging kind of way of analyzing lots of different cell types. So we're literally making billions of measurements every time we sample me.
And how often are you sampled?
Well, we sample, it's not just me anymore, it's 100 other people like me. So we sample people about every 2 to 3 months when they're healthy, and then when they get sick, we actually do about 5 additional samples as soon as they get sick, and then through a careful time series. We're trying to understand what happens when evil things happen, if you will. That is to say, if you get a viral infection or a bacterial infection. And then we also do, some people are going through dietary changes. That is to say, we have a portion of our participants who actually overeat, if you will, and they gain weight, and then they lose weight. And we see what happens when they gain weight and lose weight, and how sort of permanent that is in your molecular profile. We also have people, including me, who are going through various dietary perturbations. I'm trying different supplements to see what it does actually to my biochemistry. So then we do more dense profiling.
So Kathy told us how they're going for 1 million people, even 79,000 by the end of this year. And even that might not be representative of the entire population, which of course raises the obvious question, what is the power of one? What can you learn from one?
It turns out, yeah, it's amazing. You actually learn something interesting about every single person. So it turns out everybody's interesting, not just to their mother. Because you really will see these changes. I know exactly what happens when I eat a banana. I know what it's going to do to my sugar. I know what it's going to do to other biochemical molecules. And that's all doable. And some of that doesn't require the level of detail I just described. I wear a lot of sensor as well. So I have 6 different sensors I'm wearing right now, if you count my smartphone. So, including 3 smartwatches. So we can actually measure this kind of detail without, in that case, getting overly sophisticated. But still with current technologies, you can measure a lot of this yourself. In fact, probably many of you are. How many of you have a smartwatch, by the way? Okay, about half here, maybe a third. Okay.
So you mentioned when Stanford offered the possibility of students doing their genome a few years ago, there was a hue and cry. So you learned at least one interesting, important disease-related thing that you had no inkling of when you first did this. Tell us about that revelation and how it affected your thinking and behavior.
So it started with me sequencing my genome, because that technology was just rolling out while we were setting up the other technologies. And we sequenced my genome, and then we made disease risk predictions. And my genome largely matched what I knew about me and my family history. That is, I was at high risk for coronary artery disease. Everyone on my father's side has died of some sort of heart problem, including, we think, coronary artery disease problems. Obesity, I'm at low risk for, and nobody in my family's ever been overweight. But there were a few surprises. One is basal cell carcinoma. It came up late in the study. And it turns out, when I asked my brothers and sisters, I have 2 brothers, 3 sisters, it turns out, actually, 2 brothers and 1 sister had basal cell carcinoma. So it was in my family history, I just didn't know it. So I like to say your genome is a better record of your family history than your family is, because it actually is more precise if you know how to interpret it. Whereas family history, you don't talk about basal cell carcinoma around the dinner table, so that information doesn't transfer very well. But the other big surprise that came out very early in the study was the risk for type 2 diabetes. And because we were making all these other measurements, including my glucose had been marching along just fine, and then it actually shot through the roof. And it shot through the roof after a very nasty, it's called a respiratory syncytial virus infection. It's a common cold in kids, not as common in adults. And when I got this nasty viral infection, my sugar shot up. And it wasn't just for a few days. A lot of people say, oh, your glucose goes off for a few days. It was up for months. And in fact, I completely changed my diet, doubled my biking, started running, and it took about 8 months to bring it down to normal. So it made a big splash at the time, because it's the first time someone used their genome to predict their risk for disease, and then in my case, actually got the disease, and I was able to manage it, at least initially. Later, it turns out I'd stopped running, and we were still sampling, but nobody was looking at the data, and it went up again for actually over a year. And I only discovered it when somebody looked at it and said, Mike, you realize it's called your hemoglobin A1C. That's a measure of steady state glucose. So it was elevated. I was back to diabetic again. So there's no question I have a glucose misregulation problem. It turns out my brothers and sisters who are all thin like me have the same problem. They're all pre-diabetic. And so, yeah, and again, it was all predicted from my genome. So that's a simple case where my genome was invaluable for putting me on the alert for this disease, which in fact then I was able to catch right away. And I think that's a significant problem with healthcare today. If you think about it, when you're healthy, you go to the doctor, at least I did, about every 2 or 3 years. That's not enough, because if something evil happens in between, you know, bad things will happen in the interim, and it may be too late. You may be symptomatic, and then it's very hard to reverse. So I think that's another aspect of what we're going to want to see. I'm not even sure sampling once a year is enough to catch diseases when you need it. I think we need home tests that people will be doing every few months. And so that's what I would like to see. Since Kathy's here, I'll put in my plug. That it would be great to see frequent longitudinal sampling, not just once every blue moon when it's probably too late to do anything about it.
So I love the diabetes example because at many conferences like this or on precision medicine, personalized medicine, there's usually one speaker who asks for a show of hands from an audience that is made up in large part of people who are in the field in some way. A show of hands, who has had your genome sequenced? And at least in all the ones that I've attended, like 2 people raise their hands, and then the speaker asks, why the heck not? What is, you know, why aren't you jumping on this train? And someone will yell from the audience, because it's not actionable, because nothing I will learn will tell me anything useful about how I live my life or what I do. Obviously with some exceptions, but many of those exceptions are things that you could find out from single gene tests, BRCA1 being an obvious example. So how do you respond to the, it's not actionable, there's nothing useful I can learn, at least yet?
Yeah. So I would say, first of all, that's nonsense, because we've now sequenced a lot of healthy folks. In the first study of 12 people, 1 individual had a BRCA1 mutation, no family history of breast or ovarian cancer, no question had an inactivating mutation. Based on that, that individual opted for surgery. Actionable information, I'm not saying she had to take that course of action, but it was still very useful information to have. As we're now studying 100 people, I would say we haven't finished going over them with a fine-tooth comb. We actually spend a lot of time the way we go over genomes. It's because we do a lot of manual lookup, because a lot of the information in the public databases is wrong. About 9% of the time they put the wrong information in. Well, they'll put, this gets technical, but they'll put a probe allele in instead of the actual reference allele, so you'll get things mixed up. So when that gets cleaned up, that'll get better. But we spend a lot of manual time doing lookup, so it takes a lot of work just to analyze 100 genomes. But even from the first bunch we've analyzed, I'd say we've analyzed all 100 sort of cursory and about 20 in detail. And 3 of those individuals have actionable information. One has a mutation in a particular gene that's known to be high risk for cancer. It's called SHDB, which puts them at high frequency risk for paragangliomas. There's no family history once again, but there's a simple urine test she should do every year to see if she gets this. And there's no way she would have looked for this had we not sequenced her genome. Another individual with a protein C mutation, you know, high risk for blood clotting. Third individual who, and this is how I think a lot of this is going to roll out. This individual had their genome sequenced. We just finished it. Comes up to us and he says, my father just had a heart attack, age 61, and he realized that his uncle had died at age 53 from a heart attack, and his grandfather had died of a heart attack, also fairly young. So he said, can we look at my genome for this? Which we did, and what we found was a likely mutation in a gene that puts him at risk for dilated cardiomyopathy, so for heart attacks, essentially. So what that meant was, first of all, not only did we find the likely gene running in his family, but he had it. And based on that, he will actually be following his heart rhythms and things more than he would have had we not done all that. So I think that's how your genome information is going to be used a lot. Some of it will be actionable right then and there. That is to say, you'll see information you'll want to be on the alert for. And other aspects of it will just be held in your back pocket. So someday when something new comes up in your information profile, you'll reach into that and you'll say, well, do I have this high risk for, you know, heart attacks in my genome? And someone will actually do that analysis. So, and you'll be poised for it. So that's how I see genomics being used. What I don't understand is why people are scared of genomics when they're perfectly comfortable with family history. That is, we use family history all the time in medical practice. I think I'm preaching to the choir here anyway, right? So people are 100% comfortable with family history, physicians are, but they're scared the jabbers out of genomics. And there's no reason for that. And I think the reason they're scared is because they don't understand it. People don't prescribe what they don't understand. So we just have to get people, physicians especially, educated about all this.
Well, I think also that the American public, even the sort of scientifically, medically literate American public is in large part, they're genetic determinists. There's something about you have this gene that's associated with this that just to a lot of people feels more real, feels more fatalistic than Uncle Harold had a heart attack because maybe Uncle Harold, you know, smoked and was overweight and whatever, but somehow it's in me, I'm fated for this bad thing to happen, which leads me to the question about something else you found in your genome and how you checked with your mother about whether, and that was reassuring. Could you give us that example? Yeah, so,
well, I'll touch on two aspects of your question. So the one that Sharon raised was the issue that from my genome, I was predicted to have a high risk for aplastic anemia. And there's, if you read the literature, there's a mutation, and if you read the literature, I should have aplastic anemia, which I don't. And I was somewhat reassured when we actually sequenced my mom so we could better understand her genome. We actually sequenced my whole family. So, well, not my kids. My wife won't let me sequence my children. But anyway, we sequenced my brothers and sisters and my mom has the same mutation I do. And she's 87 now, no aplastic anemia. So that is an issue. The information may not always be 100% correct. So either it's inaccurate or it's contextual. That is to say that mutation might cause aplastic anemia in some people, but not others. And so you do have to keep on the alert. It's not guaranteed. And maybe our family is resilient to that mutation. That's possible. I don't know. But nonetheless, it will keep me on the alert. I always assumed I was fine once we sequenced my mom and saw it there, but it actually turns out, as a little technical note, the mutation's in something called telomerase, which adds sequences on the ends of your chromosome. And it turns out women, we all have our telomeres shortened, and the ends of our chromosomes shorten as we get older. And it turns out for women, that's a very narrow slope. It goes down very, very slowly. For men, it actually goes down slowly at first, but then it hockey sticks down. So even though my mom is in the clear, I may not be in the clear. I may still get aplastic anemia, but my genome has me on the alert for it, so which I otherwise wouldn't be. So I don't think that's a bad thing to know that.
So what might be your advice either to to PMI or generally to people who get their genome sequenced or exome sequenced when they do find something alarming, whether it's the Resilience Project or just the lots of studies where an association between an allele and a disease in this population doesn't replicate when you move to another continent or whatever. So whether we call it context dependent or their environmental inputs or whatever, pretty soon a lot more people are going to be getting a lot more genetic information about themselves. And going back to the poor Stanford students will be alarmed. It's not a trivial concern.
Right.
There is, James Watson famously, when he had his genome sequenced, said, "Tell me everything, but don't tell me ApoE4."
Right.
So there's a lot of potentially alarming stuff in there. So
Sure.
How might people who are obviously not as genetically informed as yourself, and God knows we don't have enough genetic counselors in this country to help them, nor do we have enough primary care physicians who probably could pass their genetics boards these days. So how should we start thinking of this information? Who should hold people's hands? What would be your advice as more and more people are going to be presented with this?
Sure. Well, you touch on a lot of different points. First of all, I would argue the power of the PMI is really to start getting good data. Having 1 million genomes with phenotypes and outcomes is going to be really, really powerful. That's what we need. Ideally, we need 1 billion. And I know George and I share the same mentality on this, and I think probably everybody in this audience does. again, I'm preaching to the choir here. I think the more data, the better, and the more open the data, the better, because it's going to be used. I would argue 1,000 open data sets are worth more than 1 million scrolled behind walls, because it won't get used as much. But anyway, I think that's the power of the PMI. We can have real data, so we'll know how often people have that exact variant I have and have this, have the disease. So we can get, everything at the end is going to be probabilistic. You're going to have a certain fraction of chance of getting something based on a genetic variant. And I think that's the information we want to get out of the PMI and other initiatives like that. As far as who should get the information and how it's returned, everyone in our study gets genetic counseled before they enter the study. It's a genetic counselor that returns the information. We talk with them in detail. It takes a lot of time about how much information they want returned. And I do think that's a personal choice. I don't think everyone necessarily wants to hear all the information, nor should they have to. That is to say, I think if you're going to get up every morning and worry about all these diseases you just heard about, that has an effect on your quality of life as well. So we don't want people, who are basically crumbling every time they wake up in the morning. They have to, feel good about themselves. So it's really what information you're prepared to handle, and I think that's where the dialogue occurs between the genetic counselor and you. Once you hear that information, you can't unhear it. So it has to be really well thought out, and people have to be prepared for the information. So once they are, then I think they're entitled to whatever information they want to hear. A lot of people want to hear if they have Huntington's, even if you can't do anything about it. Other people don't want to hear that. And I think that's the dialogue you need with your counselor, and that's the level of information. Again, for the most part, though, I think a lot of this information will be things that you'll hear about, and then you may actually adjust your lifestyle to try and reduce it. Classic cases, the common disease stuff like type 2 diabetes. If you hear you're at high risk, well, you probably should watch your diet and exercise more and such. So I think this is how you'll use that information.
So as you pointed out, genetic information is, with some exceptions, probabilistic. Your chance of getting cardiovascular disease is X% higher with this variant than with that one, et cetera. So we've been focusing on your genome, but there are lots of other omes that you have information about. So is the epigenome, is the metabolome, proteome, are sort of the next omes downstream of the
right.
genome more informative? Maybe this gene that you're carrying has been silenced and you can go on your merry way and you'll never have this disease that it's associated with.
Sure. Yeah, I actually think a genome is just a very small part of the whole equation. I don't know what fraction it is, but it's probably less than 10%. I think your epigenome is going to be just as important, possibly more important. It's one you can actually do something about. So what do I mean by epigenome? Again, maybe for those of you who don't follow this as much, your DNA can get directly modified, and so can the material that packages your DNA. And it gets modified by your lifestyle and activity. So nutrition actually affects the chemical modification of your DNA. So does exercise. There's a beautiful study by this group in Sweden. I don't know if you've seen this, where they had 23 people and they exercised one leg but not the other for 3 months. And at the end, they took out muscle from before and after, and from the exercise leg and the not exercise leg, and you could see modification of your DNA changes in one leg relative to the other. Why is that important? Well, these modifications affect the expression of your genes. And in fact, they saw that as well. So you can actually affect directly the modification of your DNA by exercise. And we still don't understand how this works very well. This is still the Stone Ages, I would argue. But this is the kind of stuff that we will learn as we do more of these studies. And by collecting this kind of information, I think collecting it on the group here and, again, millions of other people, we'll be able to better learn what the rules are. I think this is all figure-outable. That is to say, I think we will enter a world where we'll know exactly what you eat with your particular genome and epigenome, what effects it'll have. I think that is solvable in some probabilistic fashion.
Do we think, even again in the Stone Age, as you pointed out, do we think that the effect on you of eating a banana, of exercising more, of whatever, is that going to be different than in someone else who does those things?
Absolutely. We know that already. By putting glucose monitors on people, we see that everybody's different. So the way people react to different foods is different, presumably because we all have different genomes to metabolize things differently, and we all have different epigenomes, modified genomes, that probably affect how we metabolize our food and behave in other environmental circumstances. So I think everybody is unique, and I think it comes back to this power of one. I think at the end of the day, a longitudinal profile on you is much, much more important than a one time point on a bunch of, million other time points. That's actually not very useful to you, I would argue. It's how you change and what's your altered state relative to your healthy state that you're really going to want to know.
If your time and your lab members' time and everything else had been valued as what it should be, what kind of dollar figures are we talking about for a project like this?
You make sure you're sitting down for this one. We spend about $170,000 per person. Why do we, per year? Why is that? Because we're doing, the methalome is a lot of it, sorry, this is the DNA modifications. That's very, very expensive, and we go very deep. Now, we can do a lighter version of this thing for probably $1,000 or less. But we go very deep because I don't right now know what information is most valuable, so we're trying to collect everything with the idea that we'll filter out the most valuable part and then put that on a larger cohort, a larger group. But right now, because I don't know what's the most important information, we actually try and collect everything. So it's very, very expensive.
So the cost of just basic sequencing has, of course, plunged.
Right.
I can't see how the cost of smart people analyzing something is going to follow that curve, but and I think even in, President Sanders' healthcare world, not everybody is going to be able to get that kind of sequencing and analysis. So do you see any path toward which this could be available to other than the 1%?
Yeah, so don't get me wrong. What we're doing right now is a research study where we're collecting billions of data, on every person at every measurement time. By the way, on the wearable stuff, we're collecting about 2 million data points a day. So anyway, that stuff is dirt cheap, by the way, relatively speaking. So what we're doing is a research project from which we hope to extract information that can be used on a broader group. I predict that because the way the system is set up, the only people who will be able to get this will be those who pay for it, because our healthcare system, I think everyone here appreciates, is completely broken. We only really work on, we only treat people who are sick. We actually don't treat people who are healthy. And so there's no incentive to actually pay people to keep healthy. There's minor incentive, I should say. So I think what we need to do is be able to, so the people who will get these measurements while they're healthy will be the ones who pay out of pocket. That will be the wealthier people. Hopefully that will demonstrate utility, so then it becomes a normal part of healthcare, and then everybody has access. That's how I see it rolling out in a sort of two-step process. Is it the right way to go? No, but it's probably the real way it'll roll out, in my opinion.
You've mentioned wearables a couple of times. Lots of measurements. Tell us a little bit more about how that has become part of the project. It wasn't at the beginning, I don't think.
No, I think this is a pretty new technology. So we started about 2 years ago, and I use 9 devices every day. Most of them relay information directly into my smartphone, not all of them. And it's not set up properly yet to integrate all the information, but we're working on that. The ultimate goal is your smartphone will be your nexus for your health hub, if you will. We're collecting it because we're trying to correlate the biochemical measurements with the physiological ones. And along the way, we've learned some interesting things. Perhaps the most interesting one was when I got Lyme disease last summer. The way I figured it out was from my wearables. Because it turns out I had landed in Norway on an airline flight, and your oxygen tends to go down on flights and always bounces back. And when I landed in Norway, we were going there for a 2-week with my family vacation, and my oxygen went down. It didn't bounce back right, and my heart rate was elevated. I didn't realize at the time because I didn't look till later, but my skin temperature was up. And I knew something wasn't quite right, and then I had a low-grade fever for 4 straight days, and finally went to a doctor there. And I'm in Norway, where they don't have much Lyme disease. You may wonder how I got the Lyme disease. We'll come back to that in a minute. But anyway, I went to this Norwegian doctor who drew blood, said your monocytes are elevated, so you must have a bacterial or possibly a Lyme. I warned him it could be Lyme, because it didn't seem like anything I'd had before. And he was in Norway. I was a little worried he would know about this. So he was somewhat prepared for it. And then he looked at it, and he said the data, all right, I think we should give you penicillin. And I said, I think I should have doxycycline. And it was a little tense. You guys appreciate this. You're in Lyme country, except maybe Boston itself. But anyway, that's where I got it, by the way, rural Massachusetts 2 weeks earlier. So this physician, it was a little tense there. My wife will be my witness. He definitely felt I needed penicillin. I definitely felt I need doxycycline. He finally relented. I basically said, look, I'm about to go above the Arctic Circle, which is not pretty remote, and I don't want to be sick while I'm still up there. If I'm right, it'll probably still clear up. If you're wrong, if you're right, if we do what you do and you're wrong, I'll still be sick, and that's not a good situation to be in. So he finally relented. I took doxycycline, cleared it up, and when I got back, I did the Lyme test, and sure enough, it was positive. And I have the right control because I had actually given blood just before I went to Norway, so I had a sample of the blood, and it was negative then. So I had seroconverted. That's the way this test worked in that time. So we know it was definitely the case that I'd gotten Lyme. And so my wearables basically solved the story. And had I not had that, maybe I would have figured it out, maybe not. But I think that's also another important health lesson, which is at the end of the day, you're responsible for your own health. No doctor's gonna sit down and figure out my oxygen levels and my heart rate and this sort of stuff. He's simply going to say what he thinks. And some of you may have heard me say this before, but I think we're in an age of hunch-based medicine. Doctors say what they think is best based on their hunches. And we've got to get the data-driven medicine. And all you had to do is we've now gone back and looked at my data. It's very, very clear. My oxygen's low, my skin temperature's up, my heart rate's up, all figure-outable from the wearables.
We want to leave a little time at the end for audience questions, so if there's something I haven't been asking Mike, I'm going to call on you all in a few minutes. So for this audience, I think people would be interested in the fact that you post most of the information that you've gleaned where people can see it, and chime in on whether they see anything in the data that perhaps you all have missed. I'm curious whether anything interesting has just come in over the transom.
Yeah, so I get all kinds of free advice from people analyzing my data. Some of it's pretty wacky. There was one thing I thought that was interesting that we're pursuing, which is some person thought I had a mitochondrial defect, and there may be some evidence of that. My oxphos levels may not be quite what they should be. So I'm looking into that. But otherwise, yeah, I don't mind people looking over my data. As I say, I get lots of interesting information relayed back to me.
And have there been any, we've talked about health, medical consequences, beneficial ones so far. Have there been any insurance kind of consequences for you for what you've done?
Not really, and well, possibly a little bit. So when when my sugar went up, I went to my general practitioner who basically remeasured me and classified me then as diabetic. So then it was on my medical records. And at that point, we asked about increasing my life insurance and with my life insurance company, and the quote they gave me was definitely higher. But then a year later, I went to another company and asked. They said, no big deal. It was back down at the normal level. So in the end, there was no consequences. So when it did transfer to my medical record, there was a possibility that my life insurance would go up. Now, I would still argue that it's better to have that information and get my glucose under control and have my life insurance go up than run around not knowing I have a high glucose problem. So even if it hadn't come back down, I had to pay more for life insurance, I still think it would have been worth it. Again, I think there's nothing better than getting your health under control If you have to pay a little more for that, well, then it's still the right thing to do.
So you've mentioned a couple of interactions with physicians, with providers. If you could wave a magic wand, how would you change this leviathan called the American healthcare system so that it can best absorb, make use of the kind of information that you've been generating, that soon others will be generating, in a way to improve the health and the care of people? Is that even possible in the lifetime of anyone in this room?
Well, I think so, because I think people are very eager for this information, and it doesn't matter what level of education you have. I've given talks in the New Haven Library where homeless people have walked into the door and they'll start talking about their DNA, even though they don't understand it very well. I actually think this is explainable to anyone. I think it should be taught to people in probably middle school, possibly high school. They certainly shouldn't get out of high school without having some information about their DNA. Physicians definitely have to know this, and here I'll make a shameless plug, but we recently wrote a book that just came out. You can buy it for $12 at Amazon. I'm not trying to make bundles of money off this. It really is intended to get this information out to the educated layperson. It's not written to the homeless person, I'll have to say. It's written more for the educated, but it's also written for physicians. And the idea, again, is to get them comfortable with this space so they're not afraid to prescribe it. Because I think in the future, everybody will be getting their genome sequenced before birth. I really do think that's coming. And rather than try and fight it, the physicians that will do the best, same with the wearables, will be the ones that integrate this information and use it as part of their health care, not the ones who are scared of it and say, well, you know, we don't really understand that, it's not accurate. These are the things that first happened when genome sequencing came out. These days, I can't imagine having lung cancer and not getting your genome sequenced, but I can't tell you when it first came out, people were saying, well, it's not accurate enough, we don't, you know, all these excuses for why they weren't doing it. The reality is they didn't understand it. And I think that's what we've got to get rid of. So I think educating people at an early age, educating physicians now about, you know, the essence. People say, well, it's too complicated. It's not too complicated. Physicians use image information constantly. They don't understand how those images are generated, but they know how to read a report, and that's how we have to get the information back to them.
If there are any questions in the audience, just go to one of the mics if you would, and I'll call on you in a sec. Just one final question from me, Mike. It is an ongoing project, but as you look back 4+ years ago, is there anything you would have done different at the start that would have made it even better now, or has it just been evolving in a productive way? And it's all good?
Well, you know, it's always nice to add more information on, so obviously I wish I was collecting more right from the get-go, but it was quite a beast just to get the study off the ground. Even back in the early days when we were only making 40,000 measurements, that was a lot. The technologies were much more expensive, and so the amount of information you got per dollar was much less. So it's nice that things are getting cheaper. I believe in a world, and certainly we're there with the genome sequencing, where the prices will keep dropping by logs at a time. And so pretty quickly, this stuff should get inexpensive. The problem is we tend to want to collect more data when that happens. So it really never becomes a whole lot cheaper. We just collect more information as opposed to getting a lot cheaper. But maybe the clinical version will be cheaper. So
let's start over here.
Hi, thank you so much. I'm Rodrigo Martinez. I'll be a little bit provocative, because if we're not provocative here, then where are we going to be? I think I completely disagree with you when you say, I don't know why they don't get it. If we don't stop using that type of language, we keep missing opportunities to do a better job to making sure they get it. And so my question is, are you spending any of your budget in becoming better at explaining, doing better visuals, telling people that are not us, what is a genome, why does it matter, what is a gene, what is an allele? Because if we don't do that, we're going to be having discussions on this, just like the ones we're having on GMOs that we had 15 years ago that we're still having.
yeah.
So I would highly encourage us to spend a fraction of the budget in just getting better at communicating and not having this old attitude that we're still having of, well, the science, they just don't get it, and we're doing it right. When are they going to get it? Because no other industry does that. They
Yeah.
very good job at engaging people. How do we get better at that?
Yeah, that's a fair point. Now, we don't get money to do what you just said. That is to say, unless for, I guess, for talks and things. So the only way it happens, which I do, and it's another impetus for writing this book, was that I do give about, I'd say, somewhere between 6 and 8 public lectures a year. And they're often very crowded, actually. Again, because they bring in all walks of life, and I like to think I'm explaining things at a level that people can understand. So I do try and do outreach. I also do go into, we have an outreach person who goes into San Jose High School, where there's a lot of disadvantaged students there, and tries to explain what DNA is all about. They do a little CSI scene. And so we actually do a program to try to educate people pretty early. Is it enough? No, it's not enough. And I think we need to do more. And that is a budgetary issue, so I would agree. I think NSF's probably pretty better at this perhaps than some of the other groups for trying to reach folks, but I think we need to do it nationally and not just a little bit here, a little bit there. So your point's very well taken. Yeah. And I also think we're talking two kinds of people. One is getting out to the lay people, and the other is getting out to physicians, right? Physicians we want to get to right now because I want to see them not be scared of this. So somewhat two different groups, but yeah.
Question here.
So I had a question in regards to actionability. So I had an actual recent event where I discovered I had a nonsense mutation on one copy of my alcohol dehydrogenase enzyme, which A, explains why I don't deal well with alcohol, but B, also has an association with an elevated risk of Parkinson's. And so I emailed my PCP, and he said, well, avoid ETOH, which was kind of like, doctor, it hurts when I go like this, well, don't do that. But the concern I've got is we're very good at, I have elevated cholesterol, okay, take a statin. But what we're not so good at is, hey, so I have this lifetime increased risk for something that I'm not showing any symptoms from. You know, we don't seem to have a good mechanism in place for surveillance on this type of thing, or, you know, when it's not immediately actionable. And that's kind of the concern I've got with a lot of this, is where you have a 2x chance of getting something, but don't have it now. That seems to be where the gap is, and I was wondering if you could talk to
Yeah, I
that.
agree completely. Once again, it's that we're not doing things for people when they're healthy. We're not following them when they're healthy. And there should actually be, in my opinion, home measurements you would be making. They may turn out to be wearables, by the way, for Parkinson's. We'll see. But I would love to see, home tests you would do, a little finger prick test that you'd do every few months to see whether you should be on the alert. And then probably, there might be things you should be trying to do now. I don't know enough about Parkinson's. There's a lot of research going on, obviously, here. But it would be nice to follow these things earlier rather than catch them once they're symptomatic, because by then it's very hard to reverse them. So I agree completely.
Let me try one more here to even out the lines.
Sure.
Yes.
Okay, the PGP sequenced my genome, thank you. Unlike James Watson, I did find that I have one copy of the ApoE4 allele. So I study that and quite interested in that. I mentioned it to my physician, family physician, who disregarded it, said, it's genome stuff, forget about it. So of course I support the other comments here about educating the physicians about this. And are there places that people should go? I mean, I have access to a lot of information about this, so I'm not in as bad a shape as some people, but is there other things that people should do? A physician's directory of people that pay attention to genomics.
Yeah.
Probably
Is there a question on this? But I guess the answer is you know it. I don't know if now you do more mind games when you're on the airplane and things like that to try and exercise your mind more, right? That's supposed to
Sure.
stave off
diet and
Alzheimer's and
exercise.
such. So you probably play more mental games than you used to, perhaps. And I don't think that's a bad thing. I think that's where actually collecting that information could be useful. So.
Sure, thanks.
Yeah. In the case of a single allele, though, I think at least the numbers I used to have was as a European male, you have a 17% chance of getting Alzheimer's, and I think you go to 25% when you have one copy of the allele. So it is a modest increase. But
I've seen numbers the same as 1 in 6 or 1 in 7.
yeah, that's roughly the same numbers. So yeah.
So it means there's a 6 in 7 chance that I won't get a protected one.
Yeah. So I think we're in the same ballpark.
Hi. I just wanted to follow up on the conversation about education. I was really excited to hear about the work that you're doing in this area, Mike, and to hear the question because so I'm from the Personal Genetics Education Project that's based here at Harvard Medical School. I think we have a lot of common interests about raising awareness, how to engage people who are the least likely to join in this conversation, and to make it really a dialogue so that we're sharing information, but also hearing about what people think about these technologies, where they're going, how they want to or may not want to use them. So I'm happy to talk to people throughout the day about this topic. It's really great to hear it come up here.
Great. I'd like to learn more, too, about what you guys are doing. Maybe we can talk at the break. That's
Great.
terrific. I don't think we can have enough. I mean, I agree 100% with the comment made earlier.
Just an aside to the previous question, I have a domain name called Kid Omics that's up for grabs for anybody, and I believe we need to start teaching K through 12 so that the kids will graduate knowing how to deal with their omics, because they will be using the platform that we design where everyone accesses it, doctors access it, and so on. But my question is the following. Since it is precision, which is science, and medicine, and you're the omics expert, apparently, in the world, maybe, what are we doing to standardize the tests that we take that will enter the genome-wide database, and does it have to be standardized in order to do research with a capital R?
Okay.
And I will have all that done, even if I have to pay for it, but I don't know who is to delineate my genome.
Yeah.
I mean,
okay.
am I going to have Illumina do it, or Craig Venter, or I want to know from NIH PMI cohort which data collection, what is the standard for collecting data?
Okay.
Can you?
Yeah. Well, first of all, let me applaud your website and getting information out there. Again, I really think that's terrific. On the information about standardization, it depends at what level you're talking about. So if you're talking about calling the variants, that is calling the changes in your genome relative to a reference, there are some things in place. There's a long ways to go to get that more accurate and more in a more standard format. There is a reference genome out there, but that has a lot of disease alleles in it. So there's a lot of discussion about what is the right reference for a medical interpretation. And I think that's going to take a lot more discussion. A lot of this is contextual because, just as an example, you might call someone who's missing the CCR5 gene, having a mutation, but of course that person's somewhat protected from AIDS and HIV. So they're, if you will, better than normal in that circumstance. So normal is really defined contextually, and that's what makes it hard. So at the end of the day, we're going to just have a reference genome, I believe. You will call your changes relative to that, and that will lead to some risk scores in some probabilistic fashion. We will get a lot of it wrong because we have incomplete information, but I will argue it's better than random for many things, so it will be better than nothing. And I do believe it'll be better than your family history, which is what you're using now in the doctor's office. So I think it will be valuable, and I think this is why it's really, really important to have the PGP and the PMI initiatives and all these genome initiatives where the information will be shared, because I think those numbers will get much, much more accurate. And then
Right.
at the end, people can use that information into their health care. And actually, the person can decide. Somebody can say, well, you could take this drug knowing you have a 20% chance of getting something, or maybe you don't want to take it unless you know you have a 70% chance of getting it. But you could actually put the decision into the patient's hand, which is where I think it belongs.
Oh, yeah. So precision medicine on July 1 can collect people's blood, and then later, if they find that a different test is necessary, they just go to that blood and do it.
I would hope so. Sample prep is huge, and I guess this is something I'm sure Kathy knows. It is the single most important thing you do when you collect someone's sample.
Right.
And it's the reason why in our study, everybody goes to the exact same clinic, so they're all done the same. And I'm not saying it's the best, but at least I know they're all done the same, so I can compare across people really well. And so this has to become incredibly well standardized. Again, it's probably the most important thing there, and then that's correct. You can do more and more sample tests on those blood, although it probably will be a finite amount of blood. So you have to choose those tests wisely.
Thanks. And if we have genetic insurance, Gensurance is another domain waiting for one of you guys. It's a brand new form of insurance that will do omic medicine, and its job is to take all of its customers and keep them well. So it's preventive precision medicine.
Yeah. So along that line, my own belief is that insurance companies should be paying us to get our genome sequenced.
Exactly.
Because
They
you
should
actually
pay
manage
for
your
it.
health better.
Right.
Yeah. But our system's not incentivized that way, so that's the problem. But it's interesting. There are some interesting programs. You may know Geisinger, for example, is sequencing 250,000 folks by doing it in conjunction with a pharmaceutical company who helps discover. So there are creative ways of getting this done. It's not done how I would have done it, which is I would say healthcare should do it. And some are now doing it because they want to entice people into their programs too. So that maybe that'll work, especially as the price gets cheaper. Okay.
Okay.
Yeah.
Last question over at this mic.
Hi. So currently, there's the second phase of the human microbiome project that is work in progress, and I was wondering if you were part of it and thinking of extending iPOP to include like a more broad amount of people, just not only one, but do the same thing, but for the whole group.
Yeah, so on the microbiome issue, I really want to send some samples. I know Rob's here, and so I should talk to him when I saw him earlier, so that we can see how our data matches up with his and then cross-calibrate. There's no question I and other folks in our study would love to see it all compared to as broad a group as possible. As far as other iPOP groups, we're budget limited, and that's why we've gone as large as we've gone. We're actually probably a little bit exceeding our budget a bit, so that's an issue. But there are related projects popping up in the world, no pun intended. There's obviously PGP and its related things, which is very related to what we're doing. I think we do more detailed measurements than most groups. Lee Hood has launched something a little bit like this, not as deep and more limited assays. But I know there's a group in Sweden that's launching an iPOP, one in Finland now with the exact same. In fact, we've shared all our protocols with all these groups. And now one of my former postdocs is launching one in China. So there'll be these groups popping up all over the world. And my own view is that we just can't have enough of these profiles. Again, it fits exactly with the theme, I think, going on here. And the more we share this data, the better it is. And certainly I expect, I've done a data dump of my data into PGP, as George knows, and I imagine there'll be another giant one this summer. What's nice about our 100 people, I didn't say, is that everyone but 3 have agreed to make their data completely open, without PHI information. But otherwise, all that data will be out there. And I predict it'll be used as much as a 100,000 person cohort that's put behind bars, so to speak.
How do you deal with this huge amount of data? Because
How big?
resource-wise, it has to be quite hard.
I'm not sure I caught everything because of the acoustics, but there's about a half a petabyte or a petabyte of data just on me. And there'll be several petabytes of data by the end of the year on our whole cohort. So it's pretty big, but we get better and better at managing this. If we start adding imaging data, it'll get very, very large if you keep those raw files, as I'm sure you appreciate. So then I think it'll get challenging. We're fine in the few petabytes. That's not a problem these days. See what happens when it gets to exactobytes. But.
Thank you very much for the great questions, and please join me in thanking Mike.
Thank you.